pass the first sort of 2/3 of the presentation to Zoe.
Good morning, everyone. Happy to be presenting here today. So first I'll give a very brief overview. I think the past presenters highlighted the features of KAT6 syndromes very nicely. So again, just a reminder that genetic changes in the KAT6A gene cause KAT6A syndrome or related hand syndrome, and genetic variants in the KAT6B gene cause a spectrum of KAT6B syndrome, most notably SBBY, FS, and GPS syndrome. As the past presenters highlighted, there are many overlapping features between KAT6A and KAT6B syndrome, such as learning difficulties or intellectual disability, speech and motor delays, feeding difficulties, facial features, cardiac defects, and seizures. In our KAT6B patients, there's also skeletal and genital features. Very quickly, the structure of KAT6A and B genes, they are both members of the MIS family of lysine acetyltransferases. They are about 60% identical, and there are multiple different regions or what we call domains that are important for the gene and protein function. A little bit about KAT6A variant information. So a lot of our research focuses on the different genetic changes that we can see in the KAT6A and B genes. So what we know, many of these genetic changes are de novo and heterozygous. So what that means is that there are often new changes seen in children, and we all have 2 copies of our KAT6A and KAT6B genes, and one of those copies has a change. What we know is that it's presumed that the mechanism of disease is loss of function, and so again, that just means that that change in the one copy cannot compensate, and that leads to the disease presentation. What we also know is that it is that the later truncations, as the first presenters highlighted, often seem to have a worse clinical picture, and that's because there may be gain of function. And so what that means is that due to the genetic change, there's a new activity in the KAT6A or B gene. And most of you may know that there's different types of genetic changes. So we have the missense changes in the truncation. So what we mean by truncations is that there are spelling changes in the gene that change the length of the gene, and these may also be known as chrome-shift or nonsense. And then our missense variants, they are genetic changes that do not change the length of the protein, but it changes how the protein is being read in the body. And one of our the biggest significance of our study is that there's a lack of functional study for patient-specific variants, and so that's really where we come in. And we are using the fruit fly as a model to study these genetic changes. And so a little bit on why we're using fruit flies is because they're relatively low cost. We can get results very quickly. We can do a cross-in in just 10 days. We can start to see results. They're easy to genetically manipulate and create different human genetic changes in the fruit fly to study. And they share some similar neurobiology with us, actually. And what we know is that about 65% of human genes have what is called a fly ortholog or a sister gene. So this is a gene that does a similar role in the fly. And we also know that about 85% of human disease genes actually have a fly ortholog. And for KAT6A and KAT6B, the sister fly gene or fly ortholog is called ENOC. So this makes it an ideal model to study in syndromes. So one of the experiments that we do is called tissue-specific overexpression. And so what this is we actually overexpress the human gene or a human genetic variant in the fruit flies, and we observe the results of this. And we're always observing the variant to our reference. And so this could be a non-disrupted KAT6A or KAT6B gene. And so we can look at different things like, does the overexpression kill the fly? Does it do nothing to the fly? And we can also look at the different structures in the fly, for example, wings. And one of the great things about this system is that we can adjust the temperature, which adjusts the amount of expression that we're seeing in the flies, and observe subtle differences. And this is just a diagram showing that, over the past few years, these types of studies have really taken off and become a good model for studying neurodevelopmental disorders. So now on to our research hypotheses and where we kind of started this project. So the first one we have is that most of the KAT6A and B truncating variants will act as loss of function. However, some of those later truncations, the more severe clinical pictures, may be that gain of function effect, so that new activity for the gene. And our second one was that KAT6A and B missense variants will act as loss of function. And now, when we reviewed the literature, we realized really there was a lack of those patient-specific studies, and that also quite often the patients with missense variants were overlooked, and there was really virtually no studies. So that's where we come in, testing these different variants. So this is just a diagram of the 19 variants that we are testing in our fruit flies. So we have 11 KAT6A genetic changes and 8 KAT6B genetic changes, and it's a range of missense and truncating variants. They are spread out throughout the gene, so we can get a good picture of what's going on. And then we also be sure to pick hotspots. So this is genetic changes with multiple patients identified, and all of these variants are published in the literature, so we have a little bit of a clinical background on each of the patients here. So a little bit about our results, and these are complex diagrams that I will try to walk us through. So what we always want to do in our overexpression experiments is we always want to compare to reference. So I will draw your attention. I'm just going to put the laser pointer on here. Okay, perfect. So we always want to compare to a reference. So this is the human KAT6A gene without any genetic changes. So what we see in our fruit flies is that it actually kills the fruit flies, and it's toxic. It's too much protein in the fruit fly's body, which causes them to die. So what we know about, or what we can see with our truncating variants here, so this is the earliest truncation. We see that the fruit flies are actually all living. So what we hypothesize here is that this is a loss of function variant. And then what we see is this middle truncation is that the flies are not living again. And so we see that it's still a functional variant here. And then this is the later truncation here. We see that only some of the flies are still living. So this could mean that it's a partial loss of function. So not the gene is still functioning a little bit. And then what we see here, which was interesting and not as expected, is that all of our missense variants are still functional in our fruit fly model. So that's for KAT6A. So now, going on to KAT6B, and I forgot to mention before, this is overexpressing the genetic changes in all cells of the fruit fly's body. So for KAT6B, our results are a little bit different. So this time, again, we're always comparing to our KAT6B reference. And so this is the normal human gene that we're overexpressing. And we see that this time there are a few viable flies, but it is still harmful to the flies. What we see is, again, we tested truncations and missense variants. We see that most of our variants are acting as loss of function, and that's because we have the viable fruit flies. What we see here is that our last missense variant here is actually killing the flies. And so this is acting, again, differently than we were expected. So to further analyze the function of this variant, we wanted to lower the temperature. And as I said earlier, sometimes lowering that temperature can really help us understand the variants. So we lowered the temperature from 29 degrees to 25 degrees. And what we found with this later missense variant is that it was still acting differently. And so it was more harmful than the reference. And so from this, we said this could be acting potentially as a gain of function. So next thing we did is that we expressed our KAT6A reference and genetic changes in the cells that normally express the sister fly gene, so ENOC. And what we found for KAT6A is that our results were very similar to us overexpressing in all the cells of the fly. So what we saw is that the reference is still killing the flies. The early truncation is acting as a complete loss of function. We have our middle truncation still functional, and then the later truncation acting as a partial loss of function, with all the missense variants still functional. What was interesting with our KAT6B results is that when we overexpress them in the cells that express ENOC, we got a unique result from overexpressing them in all the cells. And so here we see that the majority, reference this time is actually not harmful to the flies. And so it makes it a little bit hard to interpret the other variants, but what we did find is that two of the truncations here were still functional. And again, this could indicate a gain of function for these variants. So to get a better understanding of our variants, again, we decided to overexpress all the variants in the developing region specifically. And so what we found is that when we overexpress the reference KAT6A gene, that there were some wing defects, we call them. So there was some bristle loss here, and some of the veins looked abnormal. When we overexpress KAT6B reference, and actually all of the variants, what we found is that the wing looked very similar to the control, so unfortunately we could not draw any conclusions from those, but we do have some results from overexpressing the KAT6A variants. And so these are the truncating variants here, and we see that the wings look similar to the control wing, and they're less severe than the overexpression of the KAT6A reference. And so again, this could indicate a loss of function of those variants. What was really interesting is when we overexpressed the KAT6A missense variant, we found that the wings actually were similar to the reference or worse than the reference. Again, giving us more clues that these variants could potentially gain a function. And then so we wanted to further quantify what we were seeing. So this quantification really confirmed the potential gain of function of these variants. And so what we see here is, again, we always compare to reference. So the three truncations we are seeing is that the wing size is bigger than reference, again, showing a loss of function. And then our missense variants are smaller or similar size to the reference, and there's actually one missense variant that was lethal to the flies, so it was killing the flies because their wings were so impacted. And then here, I'll just go back to this slide briefly. So when we're quantifying wings, we look at different things. We look at the bristles, we look at the veins, and we look at is there a blister. And so this is an example of a blister here. And so we quantified this, and we found that about roughly 17% of the, when we overexpress KAT6A reference, the wings had blisters. When we overexpress the 3 truncations, they either did not have blisters or a single wing had a blister. What was really interesting, again, is that our KAT6A missense variant had more blisters than our reference. So again, this is showing that potential gain of function activity. And just briefly to summarize what I've been talking about in our results so far is that we see for KAT6A for our early truncation, we're seeing that our results are all correlated to loss of function. We see that our middle truncation here has a little bit of differing results, so it could be acting as a loss or a gain of function. We're not sure yet. And this late truncation is acting as a loss of function. All of our missense variants are acting as a potential gain of function here. And then just concluding our result for the KAT6A, we see that truncations and missense are acting as loss of function. However, we do see that each of these variants could be acting as function. And I will hand it over to Dr. Marcogliese.
Thanks, Zoe. And so, you might be thinking each test in the fly might have a different result, so we have to look at all of our assays and sort of put our interpretation together. Obviously, humans, patients have one quote-unquote good copy of KAT6 and one variant. So what we will be doing next is co-expressing the reference with the variant, and hopefully that'll start to resolve some of these discrepancies. In the last 10 minutes, I'll just talk about what we have going forward as additional objectives. So our work up until date had been funded by seed funding from the KAT6 Foundation and the Canadian Rare Disease Models and Mechanisms Network. And just for everyone's knowledge, there's now been 2 Canadian families that have donated directly to the lab to sort of continue these studies under the purview of the foundation here, but for tax purposes, they can get a tax receipt by donating to our children's hospital that I'm part of. So our additional objectives are not just to assess variants that have been published in the literature, but there are a lot of people, potentially families in this room, particularly with missense variants, where their genetics report may call it a variant of unknown significance or FUS. And so we want to use these assays to start going into databases like ClinVar and assessing these variants of unknown significance. And I'll talk a little bit about what we've done looking at the fly gene itself and developing a model. However, we're scratching our heads right now because do we make a loss of function model in the fly or do we make a gain of function model in the fly to model the disease, but the reality might be different for different patients, right? And so as far as prioritizing more variants, you can see the variants that we've made, the 19 that Zoe talked about. We've now successfully made another 10 as far as cloning. 9 of them have been sent for injection. One of them, this last one, just took a lot of tries to get cloned because it was in a very repetitive region of the gene. So within the next sort of 3 to 4 months, we'll have preliminary data on how those 10 new variants are acting. One thing to keep in mind with Zoe's studies is that final exon, and one thing I just want to tell everyone and the other researchers here is right now we think that these later truncations are more clinically severe, but I do think that where those truncations are in this final exon, because it will probably be expressed because no nonsense-mediated decay, may be important, and I would love to learn from other researchers. I assume that the second last, maybe third last exon some of these might still be expressed because some of them are active gain function in our models. So the fly gene is called ENOC. It has one single sister gene to our gene paralogs. This happened through doubling of genomes during vertebrate evolution. We won't get into that, but that's just how we have two similar genes, and the flies have one. Loss of that gene, it's been well known in the fly literature that if you lose both copies of ENOC in a fly, you don't get an adult fly. We made another model called this KG4, and the model works. It's also lethal when put in trans with these other classical alleles. Our allele allows us to cross it to a reporter so we can get a pretty picture of where ENOC is expressed in the developing larval brain. So this is the third instar larval brain before they form pupa. So just think of them like any other butterfly, right? They're insects, right? But these are the optic lobes that will eventually become part of the adult eyes, which are obviously take up a large part of their brain, but they do have a central brain that gets bigger in these adults. We know from just, people may have heard of multi-omics or omics studies looking at gene expression. Well, people do this in flies, and other groups have shown that yes, ENOC is expressed in the adult neurons of the fly. I would say at least 60 to 70% of neurons. A bit less so in glia, but it is there, and there might be a role for glia in the system. So we refined, if you look at the reporting from last year, we had sort of preliminary data here, and we sort of refined it to now show that not only loss of ENOC in all cells of the fly, you don't get an adult fly. If you just reduce ENOC just in the neurons of flies, so flies have 150,000 neurons, you don't get an adult fly. If you lower the temperature of your knockdown, just meaning less, how we can titrate how much gene expression, so you have less knockdown of ENOC, you can get viable flies at 22 degrees Celsius. But at higher temperatures, they just don't exist. At 22, they emerge from pupa, but they don't live very long. Control flies can live for 60 to 80 days in a lab. None of these flies last past day 10 as adults. Also, these flies, showing you one of our eyes, they have wing defects, and it gets tricky because they actually need their wings to sort of break into their pupa. So when we see some lethality, is the lethality truly because they're dying, or just they can't break out of pupa and then die as a result of that? These flies clearly obviously can't climb as well when they're 2 days old. So that made us move into a different way of looking at this. Even though this is a neurodevelopmental disorder, why don't we just knock down ENOC in the adult flies? So we have a system where we can let flies develop normally, and then when the adults come out, we put them on food with a drug that activates our knockdown machinery. And so when we do that, we can show that these flies have climbing defects. They don't seem to have mechanically induced seizures, but my other trainee seems to think they have spontaneous seizures. We'll just need a way to assess this in flies. I did show this last year. We wanted to make a fly model, and we wanted to screen a drug or 2 or 3. And we looked at a database that shows that this drug, nilotinib, which is used for leukemia, and just a heads up, it has a ton of adverse side effects. So it's not necessarily something clinically to think about, but it's a tyrosine kinase inhibitor. Well, it was shown that at least in mouse neurons, the drug treating those neurons increases the expression of both KAT6A and KAT6B. So we thought, well, if this was loss of function in most patients, a drug that increases the expression level of the wild type or the reference, that could be good. And so as a proof of concept, we wanted to try this in our fly models. Our preliminary data, when we knock down ENOC in developing neurons and we don't get an adult fly, except for at that 22-degree temperature, we didn't do an extensive study because it clearly just wasn't rescuing at the concentration that we tested. So we moved into our adult-specific model. And we also added in luteolin because we had an email from Monica Denton who wanted us to test it. And so we ended up testing both. And so here we're just comparing. We know that this DMSO control flies, they take a while to climb. Like we don't see the true control, which takes about 5 seconds to climb. And so we're looking for these bars to go down. And it's been hard to get enough numbers, even with the adult-specific knockdowns. So these are very preliminary. There is a trend for the naloxone, and this is a low numbers, and it's both male and female flies combined. This is at day 20. And then at our day 5 results, we have more data. We still need to get to about an N of 20 in each of our groups. There's still a trend for rescue for naloxone in the males, but totally not a trend at all in the female flies. So we're unsure what that sex difference may mean for flies. They have different set of hormones and all that. So it's potentially promising, but we also may have to titrate the concentration a bit more. The other thing is just so everyone's aware, at a scientific meeting, other drugs can certainly be tested in our model that other investigators promote. So to summarize the whole presentation, what our data really supports is that we've all known for a while now, clinically, the early truncations and the late truncations are different. But as far as I'm aware, this is the first in vivo evidence in a model organism showing that they are acting differently as well. As far as I know, this is the first functional data. Hope maybe we'll learn something else new from other investigators today, that the missense variants, in our minds, at least in our fly model, are either functional, which just means they're actually not doing much to the protein, which then you can predict that the patients should be very mild if they have missense variants. But we're showing that there's some type of gain of function behavior, which in our minds looks hypermorphic because the reference shows some blisters, the missense all show more blisters. So it'll be interesting to tease that out. KAT6A seems mostly loss of function, but there's a few pieces of data, the 2 middle truncations that are acting as potentially dominant negative, actually, in one of our assays, and that one missense variant that seems to be a potential gain of function. So we're still looking at seizure-like activity in the flies, but in general, they're not really showing it. We want to finalize our drugs. One thing that we can do for KAT6A, even though we don't have a wing screen for KAT6A, because expression of KAT6A in the same cells that the fly gene expresses does not kill the fly, we can do something called humanization. So we can basically try to rescue loss of the fly gene with KAT6A reference, and then start testing KAT6A variants. Unfortunately, we cannot do that experiment with KAT6A because KAT6A kills the fly even when it's expressed in the normal cells, the fly gene. And so one thing to learn from that is that it might be dosage sensitive. So if anyone is working on gene therapies, we already know these genes, their protein products affect a lot of other genes. So it's just a caveat to think about. And the other thing we could potentially model, if we truly find gain-of-function variants, we can overexpress them just in neurons, create a model. We can test drugs in the fly, and then work with collaborators here that are working on cells or mouse models to, shore up the results with more confidence. With that, I'll take any questions. Thank you.
Thank you so much for being on board for the clear presentation. I hope this is the second of context for my question. I think that one thing that is super clear is that rare and ultra-rare for evolution is crucial. So that means that the evolution, like everybody can to protect that, is super important. So what excites me the most about what you created is that now we have the opportunity to know what are the main things we regulate if I should see. So I wonder if you have any plans to see what are the molecular fragments or the targets that are being affected with your experiment.
That's an excellent question. So with our models, we can certainly, in our different variant-specific models, we can look at something like proteomics. We can potentially do epigenetics. There's some hesitation on my part because the caveat to our model is it is overexpression, and so I would have to dive a little bit deeper in would the proteomic changes in the fly brain, right, caused by overexpressing these variants, how relevant will they translate? But it's something definitely we can do with our resources. I think we're just trying to first try to get a little more of a precise picture. I think when we co-express them together and see those results, and if that clarifies the variant impact, I think we can start doing what you're saying, express in neurons and do some of these omics technologies. We now have a spatial transcriptomics, 10x, basically all three. So physiome, xeniome. The only thing is they're not all of them are used for fly purpose of human, but we can design probes with xenium, I think, to look specifically in the fly. That's a great question.
Okay, Tom, thank you for making everyone here specifically puzzle a lot. I do like your idea of KAT6A at the same time because of their heterozygous. I think that's an interesting piece. And it'll clarify. So a lot of the work we've done in our mammalian cell models and mouse models have shown that there is an interaction and sort of that effect between the wild type copy and the regular copy. So I don't know all that data into this presentation, but I'm sure you will.
Yeah, no, I totally agree. I think because we have such a clear phenotype in the wing with the KAT6A reference, if those truly are dominant negative, the truncations, when we co-express, the wing should look normal, right? If the definition of dominant negative is poisoning the wild type copy, so we should be able to tease that out. But with the missense that are potentially gained, I don't think they're dominant negative.
Truncating was actually our dominant.
Really?
I think there's different with that, but
okay. Do you think that, because the missense seem to either be hypermorphic, potentially negative? Oh, the truncating. Yeah.
I don't think they're dominant.
The later ones.
Yeah.
Okay. So do you think they're just loss of function, or
I think they're gain of function?
Okay, so either neomorphic or okay, awesome.
I don't know exactly.
I sort of put together
Okay.
You're going to give an example, general example, not necessarily related to class, but a general example of the function and loss of function.
Yeah, that's just as an example for everyone's understanding. So you may look at our studies and you're kind of questioning why does the normal KAT6 protein kill the fly when you overexpress it? We kind of knew that would happen because the function of KAT6 is to regulate genes, and if we put in too much of it, even in the fly, the human KAT6 will affect fly genes, and doing that too much will its function will kill the fly. And so the way it's kind of a very simple assay. Well, if normal function kills the fly, if you make a DNA change to the reference, and now you have viable flies that are fine, your only conclusion is that you're losing function because the normal function when you overexpress killed the fly, and now you have viable flies. So your only conclusion is that some type of loss of function. Within loss of function, there's two options: complete loss of function or some level of partial loss of function. For gain of function, unfortunately, you can't really see gain of function in our first experiments where KAT6A reference is killing, because even if your variant causes more death, you can't see it because you have no flies originally, right? So that's why the wing is so powerful, because an example of gain of function would be, okay, KAT6A reference causes some wing defects. But if one of those variants caused no wing to be developed in a very severe wing, and we see more blisters, right? That's an example of gain of function. The complexity gets in the umbrella of the subtypes of gain of function. There's 3, and our assays can't really tease that out yet, and a bit too long to talk about the subtypes of gain of function. Yeah.
Okay, thanks. More questions?
Thank you so much for the very interesting talk. So I understand, so you're planning on doing the food session of the wild type of KAT6A in the KAT6A mutant.
No, it just in the wing, because KAT6A expressed in with our other drivers just kills the fly, so COA can't genetically do both the KAT6A reference and the variant. So just in the wing, we will co-express KAT6A reference with the KAT6A variants. We're actually going to try expressing KAT6A reference that causes the wing phenotype and test the KAT6B co-expressed with it, because we want to check if KAT6B modifies.
Yeah, so I was wondering whether you are also planning on trying the food session of B with A.
That's exactly what I just said. Yeah. So yes, we're going to do that. We have no idea what it'll show, but I don't know if KAT6A and KAT6B can interact. But if they can, it might help us figure out how those KAT6B variants act.
And if I'm understanding correctly, they're also planning on the food session of the wild type copy of KAT6B.
Sorry, say that once more.
Food session code KAT6B wild type as well as the
Yeah, we would actually have to make a fly that has KAT6A reference with KAT6B reference, and all the variants for sure. Yeah. On the list. Thanks.
Thank you. Yeah, questions about the work of.
Thank you.
Okay, thank you very much.